Hey guys, so we are on the nervous system today and we will talk about nervous tissue. I'm sorry if I sound weird, I'm sick and I'm using a different mic right now. And I'm in the middle of a hallway, so if things get loud, I'm sorry but this is after school, don't worry. Anyway, so first let's introduce you to the nervous system. It's made up of two types of cells. So we're first going to talk about nervous tissue. So it's made up of two types of cells, neurons and neuroglia. You can see them in this picture. So the neurons are actually so the neurons are all over the place, and then there's neuroglia all over the place as well. Anyway, what's so neurons? They're made up of three principal parts: dendrites, cell body, and axon. Initial body is an principal part, but it's an organelle. It's basically the rough ER of a neuron. So people refer to missile bodies, you know what it is now, and they can be classified. Neurons can be classified into two specific ways. They can be either classified by structure or by their function, and we'll talk about this in a moment. So right now we have, right here we have a regular neuron, a motor neuron specifically, and as you can see, there are a bunch of dendrites. These take in electrical signals from other neurons. And then after that, they're processed in the synapse and are propagated down the axon. The axon is this little tail, and it sends signals to the other neurons. So at the end of the axon, there are a bunch of things called axon terminals, and at their ends, they have these little knobs called synaptic knobs or terminal boutons. Um, these axon terminals are also called telodendria. And so there are also these things called myelin sheaths around a neuron, and myelin is responsible for making the action potential more efficient, and we'll talk about this in a moment. And then here's an axon hillock, which is the area where the axon and the cell body meet. So now that we're done with that, I'll talk about the different types of neurons. So here's how they're classified when you're classifying them by their function. So this blue neuron right here is a is an efferent neuron or an afferent, because it's sending signals to the central nervous system, which is made up of the brain and spinal cord. We'll talk about that next video. But this purple neuron right here is called an interneuron, and it sends signals from the afferent neuron to what is called an efferent neuron, and an efferent neuron is this red neuron right here. It sends signals to other parts of the body, and those other parts of the body are called effector organs. And so in this case, the effector organ is this muscle right here. And you might have seen this you might know what this reflex is. It's more commonly known as the knee-jerk reflex. However, to be more specific, it's actually called the myotatic reflex, where the doctor hits your knee and then after that you kick. So this is what happens. So now let's talk about basic neuron types. So there are actually three basic neuron types. This pyramidal cell, it's an important mention, but we actually won't go over it that much. So basic neuron types: bipolar, unipolar, and multipolar. So bipolar looks like this. So one end has the dendrites and one end has the axon. So then after that, here's a unipolar neuron. So it has one string instead of like two sides. The whole thing is on one side. So this side is the dendrites. Are the dendrites, and this side is the no, yeah, this side is the dendrites, and this side is the axon terminals, telodendria. There are also pseudo unipolar, but I actually won't go into that. And there's multipolar, and this is the most common. You usually see these two in sensory areas like smell, taste, special sensory areas, but they're not really very common. But these are very common. These are motor neuron. Well, they can be motor neurons. They're multipolar neurons, so they have many dendrites. And as a general rule, well, actually as a rule anyway, they only neurons only have one axon, but this one has many dendrites. So now let's talk about helpful terms. Nerve fiber means axon. Nerve means a cluster of different fibers in the peripheral nervous system, and we'll see their layers in a moment. And then there's nerve tracts, and those are clusters of fibers in the CNS. So take a look at this picture. Here's a nerve, a peripheral nerve, and so right here you can see the epineurium, which surrounds the whole nerve. And then after that, they're in little fascicles, and the outer layer of that fascicle is called the perineurium. And then after that, there are more sheaths, I guess so to speak, not necessarily these myelin sheaths, but the these sheaths right here, and those are the endoneurium, and those surround axons. So now let's talk about neuroglia. So there are different neuroglia depending on where you are talking, where you are in the nervous system. So they're in the central nervous system, which once again consists of the brain and spinal cord. There are astrocytes, oligodendrocytes, microglia, and ependymal cells. In the peripheral nervous system, there are two different types of neuroglia, and the peripheral nervous system is the spinal and cranial nerves, and those are the Schwann cells and the satellite cells. Schwann cells are also called neurolemmacytes. Anyway, so right here, as you can see, there's an astrocyte, and it's called this because it has many protoplasmic extensions which make it look like a star. They're hence astrocyte. These are responsible for maintaining ionic balance within the nervous system. Then there's the microglial cell right here. So your microglial cell is basically like a phagocyte within your nervous system. It's a special phagocyte because immune system cells can go all over the place, but they can't really go into the nervous system because the nervous system is very fragile and very important. Anyway, well, all systems are very important, but you know what I mean. So then there's ependymal cells right here. As you can see, these are usually in fluid-filled cavities, and they have cilia. A lot of them have cilia, not necessarily all, but a lot of them do. And the cilia circulates cerebrospinal fluid. And right here we have an oligodendrocyte, and these make myelin sheaths, and myelin sheaths actually insulate the cell, um, and they help with saltatory conduction, which we'll talk about. It's something important to the action potential. And here's the peripheral nervous system. So it would be a cell body right here with satellite cells surrounding the cell body. And then after that, right here, we have myelin cells. I forgot to mention earlier when we were talking about the anatomy of a neuron that there are little spaces between these myelin sheaths, and these are called nodes of Ralphier, also called nodes of Ranvier. Depends on what you want to make it sound like, but I think Ralphier sounds cooler. So, anyway, now let's talk about white matter and gray matter. Just to clarify a few things, white matter is the myelinated parts of the axon, gray matter is the unmyelinated parts of the axon. Not axon, the neuron. So, right here we have a multipolar neuron. So right here, you can see this part is myelinated. So this part would be considered white matter, and this part would be considered gray matter, and this part as well, since it doesn't have myelin. So anyway, impulses of nerves. So before I talk about this, I recommend that you watch the video in chapter 1 about the resting membrane potential. But if you feel good with that, that's great. So now let's start. Graded potential, it's changes that happen in a cell membrane for a short distance. So what happens is a stimulus occurs, and that will open channels, but it won't open the next channel. It won't propagate. It'll just open channels. Sodium will come in, but it'll just but the sodium will just propagate down, but not the sodium channels opening, propagating. If you know what that means. But anyway, if you know what I mean. And then after that, there's action potentials, and those go for long distances. So let's talk about action potentials. So it starts off with a graded potential, actually. A stimuli causes a graded potential, and the amount of sodium enters in. But when the amount of sodium enters in so that the membrane potential becomes around negative 40 to negative 55 millivolts, or negative 55 to negative 40 millivolts, that's called the threshold, and that's when an action potential starts. And once that action potential starts, you cannot stop it. It's either yes or no go. And that opens sodium channels, and the action potential will propagate, and it'll open other sodium channels, adjacent sodium channels. Then after that, repolarization occurs. So sodium channels will close, but the potassium channels will open, and that will allow calcium to leave, go down its concentration gradient. But the problem is that even though it's set to normal, it's not necessarily where it should be, because your electrolytes will be out of balance. Anyway, so right here we have a picture of an action potential. So here's a resting potential, and right here would be failed initiations. These would be graded potentials. But then a graded potential would start an action potential, and once it reaches the threshold, it'll propagate down, and then after that it'll release its neurotransmitters, and then after that sodium channels will open or close, and the potassium channels will close, and that will allow it to go back down. However, it'll undershoot a little bit, and that is called hyperpolarization. And here is where something called the refractory period occurs, and that's when they get your electrolytes back to normal via the sodium-potassium pump. So this is how it ideally would look like. You'll see this in textbooks, but this is how it should look like. Anyway, so here's a little more of a visual picture. So what'll happen is neurotransmitters go onto the cell, go onto the dendrites, and that's processed here. And then action potential will occur, and that will open sodium channel. And once the threshold is reached, which is around negative 55 millivolts, it'll allow for an action potential to occur to propagate down a membrane. Now remember when I was talking about nodes of Ranvier here. So what happens is right here the action potentials only happen in nodes of Ranvier. So what'll happen, or Ranvier. So what'll happen is they'll go in here, an action potential will occur, but once it gets to the node of Ranvier, it'll just be a graded potential. It'll go for a short distance, and then after that, there's the node of Ranvier again. There's the action potential short distance, then action potential, and then after that, the sodium will just go short distance, changing the membrane. But anyway, so now that we're done with that, the synapse. So synapses are basically the gaps between presynaptic and postsynaptic cells, and that's where the impulses pass. So calcium channels. So what happens in a synapse is that calcium channels open, and that will calcium channels open at the end of an axon, and that will cause vesicles to release neurotransmitters. And depending on the transmitter, it can be excitatory or inhibitory. So it will either start another action potential or stop another action potential, respectively, and that's called excitatory postsynaptic potential and inhibitory postsynaptic potential, respectively. And then after that, the neurotransmitter is either recycled or destroyed. So here's a little picture. So the nerve impulse goes down this way. Then after that, calcium channels will open when it reaches the end of the axon, and that will stimulate these vesicles to release neurotransmitter, and the neurotransmitter will go on to the cell. I'm done for now, I'm done with this part of the nervous system. Thank you for watching, and see you next time.